Okay, good afternoon, everyone. I hope that you are doing well. How is everybody today?
Doing good. How about yourself?
I'm okay.
Good, I'm glad to hear that. I'm getting a little stir crazy. I'm tied up with a 6-year-old most days, but other than that, I'm doing okay, and I hope you guys are as well. Okay, um, so let's see here. We're going to have to finish chapter 13 today. Let me just make a couple comments. Believe it or not, including today, you have 3 weeks of lecture remaining and then final exam start. We're really not that far away from the end of the semester, which I think is probably a good thing. Everybody really needs a break. Everybody needs some rest. So I'm going to ask you to try to stay as focused as you can for the next 3 and 4 weeks so that you can finish the semester strong and get your money's worth out of the semester that you already paid in and are working so hard towards, okay? So next Monday is exam 3, okay, a week from today. That means in this afternoon's email, I will be releasing the study guide for exam 3. So that will be coming out this afternoon to you, okay? What we need to do is finish chapter 13 today. Any time remaining I'll spend on chapter 14, and then on Wednesday we will spend that on chapter 14, okay, because chapter 14 is on exam 3. So things are starting to get compressed a little bit, and you will find me picking and choosing specific topics that I know I need to cover with you so that you have some support for your upcoming lecture exams. So you have your lecture exam coming up 1 week from today. You have your second lab exam coming up 2 weeks from today. And for those that have perhaps missed, remember, there will be a second lab exam, and it simply will be on the reading of the lab manual. I won't ask you to do anything practical because that would be impossible because we didn't do it. But on the reading of the lab manual, there's some basic ideas and knowledge that I can expect you to read and understand on your own. These sorts of things will be tested, and I will be giving out a study guide next week for the lab exam. So before we get started, do you have any questions at this time? All right, gang, let's get into it then. We're in chapter 13, which is the body defense chapter. We got the first and second lines of defense covered very, very nicely in your notes, but we really only got started with the third line of defense, which is the immune system. Okay, you're not going to be an expert on the immune system from one lecture in this course. It is a very deep topic. My hope is that I can give you a pretty sound overview of what happens in your body when your body tries to protect you. Okay, so, all right. What I'm going to do is go back to the screen share, okay, and at the end of last class, let's see here. At the end of last class, we had answered this question. What does it mean for a B cell to identify a threat? What is actually happening? B cells must actually touch the threat. There must be an interaction between a receptor on the surface of the B cell and a molecule on the surface of the pathogen. Let me get the chat window up here so you folks can get a hold of me if you need to. Okay. All right, gang. So here's where we're going to pick up. There's lots to do. I could spend another week on this topic with you. I mean, it's a fascinating topic, but we just don't have time to do that. So what I'm going to have to do is talk to you in a way that gets the basic idea through. So right now, what I'm going to do is give you an example of how the immune system can work in your body. So this would be an example called Steps in an Immune Response. Steps in an immune response. We're going to walk through it together, how your body actually tries to protect itself. Okay? And so, let's see here. I really prefer to do this by drawing some cartoons out. It's just better to understand than trying to write a bunch of words down. So I'll go back and forth between the Word document and the PowerPoint to explain this. Okay, so here we have the PowerPoint, and this again is to illustrate steps in an atypical immune response. So what you will want to do is give yourself enough space on your note-taking page, maybe even half a page or more, because we're going to write down a giant human cell. Okay, so at this point, right here we have a cell, and I hope you recall from last time I did show you a video on the immune response, and I sent you a YouTube link in last week's email to that video. It's a good starting point to begin to conceptualize what we are talking about here. Okay, so in your body, there are defense cells that can protect you. And so for our purposes of conversation right now, we'll say that this is a macrophage. Okay, so we have a macrophage. Okay, so typically what happens here in the steps of the immune response is that something foreign enters your body. I mean, let's be honest. Right now, that is definitely happening out in society because people are dealing with the coronavirus. So I'll use a virus today as a typical example. Okay, so typically what will happen is we'll have a virus. Enter the body. Okay, so let me do this as step A. Okay, I'm going to go back and forth between the Microsoft Word document and the PowerPoint presentation. Okay, so letter A, I'm going to reference in my notes in the Microsoft Word document. Okay, here we go. Okay, letter A. So letter A is that something foreign enters the body. That something foreign, in our example, is a virus, but it could in reality be a bacterial cell, okay, a bacterium. It could be a fungal cell, okay, so it could be fungi. If you've ever had athlete's foot, that's caused by a fungal infection. There are forms of microbes called protozoa that can cause us problems. Okay, there's a variety of different microorganisms that can cause us problems. All right, so today we're using virus. So something foreign enters the body. Okay. All right, now the macrophage is one of our defense cells. In fact, it's a second line defense cell if you go back and review your notes. You can think of a macrophage as sort of a police officer, right? You know, now there's lots, you know, we make jokes about police officers eating donuts and things like that. But if we really think about it, you know, at least one thing that police officers do is typically drive around our neighborhoods and communities looking for trouble that might be springing up and hoping to help out and prevent trouble before it occurs. So think of a macrophage as a police officer that's floating through your bloodstream and other body fluids of the body. So what's going to happen is the macrophage encounters this virus, right? The macrophage is very near the virus, And the macrophage suspects that maybe something isn't right here. So like a police officer going up to a suspect and patting the suspect down, the macrophage is going to go up to the virus and it's going to start to touch it, right? We talked about last time that the immune system has to touch things. So the macrophage is going to touch the virus. It's going to pat it down like a police officer patting down a potential suspect. And that macrophage is looking for something. That macrophage is looking for identification that would indicate that the virus belongs to the body. Now, the macrophage isn't going to find that identification because the virus does not belong to the body. But here's a question coming through the chat window. What type of molecule will the macrophage look for when it pats down the virus? Can you answer that? Do you know that from your reading? What type of molecule will the macrophage look for when it pats down the virus? An antigen. Okay, Ellen, that is not a bad answer. You are definitely in the ballpark, but I need a little bit more specific information. What kind of antigen? So gang, what kind of molecule, which is an antigen, specifies to the body's defense cells that other cells belong in the body? I'm looking for a self-identifying molecule, a self-identifying antigen. Do you know what that is? Okay. All right. I'm hearing crickets outside. Okay. So I see a white blood cell response, and I appreciate the participation, but that's not it either. Okay. So let's go back to the Microsoft Word document. Letter B, the macrophage, which is kind of like our police officer. Okay, encounters the virus and begins to physically inspect the virus, right? This is the pat down. Well, the macrophage, folks, is looking for a very special category of molecule that, if you read chapter 13 carefully, it is in your textbook. It's called the major histocompatibility complex, MHC for short. Macrophage is looking for major histocompatibility complex, MHC proteins. Okay. Let's get that underlined because that's a very important vocab word. Okay, macrophage is looking for major histocompatibility complex proteins. All right, these are proteins that identify cells as belonging to your own body. In other words, these are self-identifying antigens. Okay. All right. I mean, you think about the identifications we carry around in our wallet. We have identifications that probably tell people that you are a citizen of the state of Pennsylvania and you are licensed to drive a motor vehicle in Pennsylvania, right? Now, some of you might have a license from New York or from Maryland or who knows where, maybe from Europe. But the fact of the matter is we have different kinds of identification that we walk around with. The cells of our body also have these molecules that identify them as being part of the own part of the community of cells that make up your own natural healthy body. Now, a virus, like in our example, it's not going to have these MHC proteins because it doesn't belong in the body, and it came from outside the body and infected our cells, right? So it doesn't have this. Okay, so let's continue here. Let me stop. Do you have any questions right now? All right, so we have a macrophage that has encountered this virus in the body. It has tried to ask for identification. It's patted the virus down. It did not get a good answer. That's like the suspect ignoring the police officer. The macrophage did not find any major histocompatibility complex protein. So, you know, if a police officer asks you for identification and you don't provide it, there's a good chance you're going to get arrested and sent to the police station, right? Well, it's even worse for a potential virus or pathogen. So the macrophage did not find any MHC. That really alarms the macrophage that something is wrong here, that we have an intruder in the environment. So the next thing the macrophage is going to do is literally ingest the virus and eat it. That would be like the police officer eating the suspect. So the macrophage gets concerned, and you know, I'm speaking metaphorically right now to make the story interesting. Obviously, the cell, you know, is not a human being, it's simply a cell, but the macrophage gets concerned, and so the macrophage eats the virus. This is the process of phagocytosis that you've read about in your cell, in your book. Okay, so the macrophage eats the virus. It internalizes the virus. All right, so what we're going to have here at this point is that the virus is brought into the cell. Okay. The virus is brought into the cell. Now, in a situation that's happening in the real world with like coronavirus and things, you know, the coronavirus is brought into the cell, and unfortunately, the coronavirus is able to bypass the cell's own natural defenses, and that's something I could talk to you about if this was a microbiology class, but we don't quite have time for that. Okay, so what's going to happen next is the virus is brought into the cell. All right. And the cell is going to use its own cellular anatomy to defend itself. Can you please type into the chat window, what organelle do you think the cell is going to use to chew the virus up? What organelle, what part of the cell is the cell going to use to chew the virus up? You should be able to answer that. Okay, I see I'm getting you guys today, okay? What part of the cell will chew the virus up into pieces? Okay, thank you, Riley. Nicely done. Yes, that is in fact the lysosome, right? So we have the lysosome here, which you remember is the recycling facility of the cell. That virus will be passed through the lysosome, and what will be spit out are pieces of the original virus. So we will have, you know, fragments, kind of weird shaped fragments of the original virus. So these are viral fragments. Okay. So the lysosome will be used to digest the virus. let me put that into your notes at this point. Let's see here. So let's see. Step C was phagocytosis. All right, so step C is when the virus was brought in, and that's in your notes. Okay, we'll make this step D here. Step D. The macrophage uses its lysosomes to eat the virus and break it into pieces. Okay. So one take-home message at this moment is you see that even though it's kind of laborious and tedious and annoying to do all this memorization in the course, that if you can't remember what a lysosome is from chapter 3, then it makes this conversation we're having about body defense much harder to understand. That's why it is a necessary evil that you memorize some things. All right, so the macrophage uses its lysosome to eat the virus and break it into pieces. At this point, this specific virus is destroyed and it is no longer a threat to the body. Unfortunately, when a virus infects you, it's very unlikely you're only going to get infected with one virus particle. You're probably going to get infected with 5 million virus particles, right? When, if you are unlucky enough to be around someone that's shedding coronavirus, if you're breathing in that air, every time you breathe in that air, you're breathing in millions of coronavirus particles, and that's the problem. So while this one cell has destroyed this one virus, there's still millions of other virus particles floating around in the body. So what this macrophage is going to do next? Okay, any questions? All right, so what will happen next is that this macrophage is going to use its rough endoplasmic reticulum, which you also learned about. It's going to use its rough endoplasmic reticulum to manufacture a category of protein which I just alluded to, which is MHC. Okay, so let's see here. How many different colors can I get on the screen at one time? Let's do this. No, let's do let's do this. Okay. All right. So the rough ER of the cell is going to be used to make MHC proteins. Okay. So these straight lines are MHC proteins. And what the cell is going to do is it is going to take these digested viral fragments, which I'm going to circle in orange at this moment. It's going to take these fragments and it's going to bring them down and it's literally going to glue them onto the MHC proteins. So we're going to glue these viral fragments onto the MHC proteins. All right, so we've got a piece of virus here like this, okay. All right, so these viral fragments are attached to the MHC proteins. All right, so let's make activity at the ER step E. All right, and let's go back to our notes and talk about that. All right, E. The macrophage uses its rough endoplasmic reticulum, okay, the rough ER, to manufacture fresh MHC, right? This MHC that I'm highlighting right now on your screen, to make fresh MHC protein. The digested viral fragments are glued or attached, if you will, attached to the new MHC proteins. Okay. If you're following along at home, I'm going to type a question into the chat window. What is the next thing the macrophage will do with these MHC viral fusion molecules? Okay, what is the next thing the macrophage will do with these MHC viral fusion molecules? Please type that into the chat window if you know an answer. Okay, gang. So, I know things are difficult right now, but remember, you really do have to focus at home. You really do have to read this content. Anybody? Okay, well, let me help you out. Here's what's going to happen. I'll go back to the PowerPoint. These viral fusion proteins here, what I'm highlighting in orange right now, okay? What the cell is going to do is going to take these new molecules and it's going to stick them out on the surface of the plasma membrane. Okay, so we're going to take this cargo, and we're going to put them outside the surface of the plasma membrane. Okay, I'm not the best at drawing, as you are well aware, but you get the idea that that they're going to be stuck outside the plasma membrane of the macrophage. Can you tell me, why is the macrophage doing this? Okay, in our conversation together right now, we're saying that the macrophage is taking these MHC proteins, which it has attached viral pieces to, and it's sticking it outside the plasma membrane. Why is it doing that? Anybody? Is everybody passed out on their living room couch? Okay, Kirsten, to protect the nucleus of the cells. Thank you for trying. That's an interesting idea, but no, that's not really it, but thank you for trying. Anybody else? Okay, gang, when the macrophage does this, okay, when the macrophage takes these MHC proteins and sticks them outside the plasma membrane, this is a process that you should be reading in your chapter called antigen presentation. So the reason the macrophage is doing this is because the macrophage wants to present these pieces of the viral pathogen to other defense cells to see if the other defense cells have ever seen this virus before. So the whole reason for doing this, okay, if the best analogy I can think of is this. Imagine that the MHC viral proteins here that I'm circling with my cursor right now is the same thing as a sketch that a sketch artist made of a crime victim's description. And what this cell wants to do is take the sketch that the sketch artist has made and show it around to people in the community to see if other people in the community have seen the perpetrator, to see if they have seen the burglar, to see if they have seen the virus. that is the whole purpose of why the macrophage is doing this. Okay. Another question. Am I making sense? Okay. What can I help you with at this time? Okay. All right. Folks, I do appreciate the feedback, and I need the feedback because I don't know if you can't hear me because the microphone is cut out. I don't know if you're not understanding. I need you to give me feedback so I know how to help you. Okay. So I will continue here, and let's make this part right here letter F. Okay. And let's go back to our notes. All right, so letter E, we said that the macrophage made MHC. The digested fragments are glued and attached to the new MHC proteins. And letter F, the MHC viral fusion molecules are placed outside the plasma membrane of the macrophage. Okay. Now, when this occurs, the macrophage is doing something that's referred to as antigen presentation, meaning the macrophage has now become an antigen presenting cell. Okay. Macrophage has now become an antigen presenting cell. And again, the whole purpose of this is for the macrophage to act like a police officer carrying around a sketch, a photograph, a picture of a suspect around the community, asking various people, have you seen this virus before? Have you seen this burglar before? The purpose of this behavior is to show a picture or sketch of the virus, of the burglar, to surrounding cells to see if they have ever seen the virus. Okay. Questions at this time? All right. So now, you know, in the analogy of a police officer roaming around looking for somebody that's seen the burglar, you know, what's happening here is that this macrophage is roaming around the police station. Okay, in the body, in the analogy that I'm trying to use with you, what would be the police station? What is the police station? Another way of asking the same thing is, where is the macrophage showing the sketch at this time? Yes, the macrophage encountered the virus someplace in the body. But when the macrophage ate the virus, and started processing it, the macrophage started to move to the police station. Where is the police station in our body? Where are you going to show a crime victim a notebook full of photographs? Well, the police station. Where is the police station in our analogy? Okay, folks, dig deep and okay, thank you, Maddie. The brain. The reality of it is that this showing off the sketch could happen at any location of the body. But the location that we have the most likelihood of success is actually going to be in your body's equivalent of the police station, which I know you know the answer to, and these police stations are called lymph nodes. So the macrophage, which has digested the virus and put pieces of the virus out on its surface, between locating the virus and presenting the antigens, the macrophage has physically traveled into the lymph node to ask other fellow police officers if they have ever seen this virus. Okay, so Roman numeral number 2 here, the purpose of this behavior is to show a sketch of the virus to surrounding cells to see if they have ever seen this virus. Okay. Showing the picture is usually most productive in the lymph nodes. Okay, in the lymph nodes. Because it's in the lymph nodes where we have our professional police officers, right? I mean, think about it this way. It is technically true that any one of us as a citizen of the United States, technically, we can make a citizen's arrest. But when's the last time you ever heard of anybody actually doing that? I mean, most of us don't actually make a citizen's arrest, right? We rely on the police, which are professionally trained people, to arrest people that are doing bad things. Well, the professional police officers are in the lymph nodes. What is the name of my professional police officers? What are the professional police officers? What do I call them? I mean, we're calling a macrophage a police officer, and that's okay for our purposes. But there's two other kinds of cells that are even more professionally trained to do this work. Do you know what they are called? We have talked about them online last week. Thank you, Kirsten. Phagocytes. Again, I really appreciate the feedback. That is not correct, but you tried. Okay, let me give you a hint. What kind of defense cells are born in the bone marrow? One of them is trained in the bone marrow, and the other goes to the thymus for training. What are the names of those kinds of cells? All right, well, you might recall that I'm talking about the B lymphocytes and the T lymphocytes. So what I'm saying is the macrophage that's showing around the photograph of the virus has actually moved into the lymph node and is asking the B and T lymphocytes if they have ever seen this virus. Okay, so in your notes, we will put that Roman numeral 3, showing the picture is usually most productive in the lymph nodes where B and T lymphocytes are present in abundance. Okay, It is possible that the B and T lymphocytes could be floating around your body fluids, including your bloodstream, but the majority of them are going to be in the lymph nodes throughout your body. Okay, so let's go back to the PowerPoint. Okay, so we have this defense cell, which has eaten a virus, destroyed it into fragments, attached those fragments to MHC protein, placed that cargo out on the surface of the plasma membrane. And at this point, this activity here is happening primarily in the lymph nodes. And what we need is to get a defense cell. What we need is to identify a defense cell that is capable of recognizing the virus, meaning we need a defense cell that has a receptor capable of bonding to one of these viral fragments. So as an example, let's see here. Let me put two defense cells up here. Let's say I have defense cell A. Defense cell A, which has this receptor, and defense cell B, which has this receptor. I'm not very good at drawing. Let me modify something here. Let me make this receptor a little bit more ridiculous. Okay, there we go. All right, gang. For those still watching and thinking here at home, is it defense cell A or B that has a receptor that can match one of the viral fragments shown at step F? Which one of these? Okay, all right, Maddie, Riley, great. Okay, wonderful. Do you see that it is in fact B? In other words, okay, you notice that this receptor here has the right shape that it could dock with this shaped receptor, excuse me, this receptor here that's circled in orange has the right shape to dock with this piece of viral fragment that looks like a square. And that's what I'm trying to get you to see. We need to find a defense cell, a lymphocyte that has the right receptor corresponding to the proper shape of that molecule being presented. And for reasons that I kind of don't have time to go into right now, the kind of defense cell that we really want to get, the attention of, is actually a helper T cell. All right, so these are going to be T subscript H cells, helper T cells. All right, so let me make this in the drawing. Let me make this letter G in our notes. Okay, this is letter G in our notes. All right, let's go back to the Microsoft Word document. Okay, letter G. If and when. Okay, and notice I'm capitalizing those words. If and when a helper T cell, so T subscript H, right? A helper T cell, T subscript H, when a helper T cell with a receptor of the correct shape is able to bind to a presented fragment of the original virus on the surface of the macrophage. If and when that happens, your immune system turns on. Okay? Everything we've been talking about so far has been under the context of the immune system not really responding in a specific way to the virus that's threatening your body. To get the immune system to respond in a specific way to the virus that's threatening your body, we need to locate a helper T cell with the correct receptor capable of binding the pathogen. And that is achieved through this antigen presenting process that we've highlighted up here in step F. So, and I will go back to the notes in just a moment. When this tuning fork is able to dock here, when that thing actually binds, we have the activation of the immune system. It's a very powerful moment, if you will. Okay, before I go any further, any questions? All right, okay. So let's continue then. From this point forward, I think I can describe what I need to describe primarily in the Word document. So I'll stick with Word for a little while at this point. Okay, so let's assume, well, question that I need to put into your notes here. What are the chances of locating a helper T cell with the correctly shaped receptor capable of binding a virus that the body has never seen before? What are the chances of that happening? What are the chances of locating a helper T cell with a correctly shaped receptor capable of binding a virus that the body has never seen before? I mean, you folks have a general idea that if you get sick with a microbe this year, if you were to get sick with the same microbe next year, if you were to be exposed to the same microbe next year, you probably wouldn't get nearly as sick because you would have immunological memory. But in circumstances where the body has never ever seen the pathogen, in this case a virus, now how can your body possibly have a receptor to something it's never seen before? How does that work? Any thoughts? All right, before I give you the answer to this question, I want to, Alexi, you say making one. Well, Alexi, how does the cell know what to make? Okay, I appreciate the feedback. It tells me that you are in the game here, and you need to be in the game by looking at the virus protein. Okay, Alexi, if it looks at the virus protein, it won't yet know. The cells of the body won't know that it's a viral protein yet. It won't know that yet. Let me lead you down a slightly different way of thinking about this, but thank you for trying and thinking about this. That's important. Let me ask this. If you wanted to win the lottery. You know, some of you have probably played the Powerball lottery in Pennsylvania here. If you wanted to win the Powerball lottery, and you had enough money, how could you guarantee yourself to win the lottery? How could you guarantee, how could you make it certain that you're going to win that lottery? So the next lottery drawing for Powerball is Wednesday night. How do you, how are you going to guarantee that you win the lottery on Wednesday? How are you going to do that? You can't cheat, you can't steal, you can't bribe anybody, okay? So what's left? If you need to win the Powerball lottery on Wednesday night, what could you possibly do to ensure that you win? Okay, Alexi, there it is. Buy all the tickets. Now keep going with that, Alexi. What's true about all, what do you mean by the statement, buy all the tickets? Can you expand upon that, Alexi? Can anybody expand on what Alexi's trying to tell us? He said, buy all the tickets. What does he mean by that?
I don't know. I can't write in the messenger, but the more tickets you buy is like the higher chance you have of winning.
So who am I talking to?
Shateria.
Shateria, stay with me. So,
Okay.
Shateria, do you mean that I need to buy 6 tickets that have the same number sequence?
No, different number sequences.
Okay, that's what I'm trying to get out of you. Thank you very much. Okay, so what we need to do to ensure we win the lottery is buy a group of different lottery tickets which have different numbers on. In other words, you need to buy so many lottery tickets that you buy all the possible number combinations that can be produced in the lottery drawing. Does that make sense? Okay. Purchase enough tickets that all the possible lottery ticket number combinations are represented. Now, gang, that's insane. I mean, you would spend more money buying tickets than the amount of money you would win in the lottery drawing itself. So that in the real world is not a very good pragmatic strategy for winning the lottery. But I hope you see it is mathematically valid. You could buy, if you had enough money, all the lottery tickets with all the different possible number combinations that you can draw in a lottery drawing. And if you did that, you would definitely win the lottery. So in an analogous fashion, I come back to my original question. What are the chances of locating a helper T cell with the correctly shaped receptor? Well, the answer is very good, because before you are infected, your immune system is preparing to win the lottery by manufacturing many B and T lymphocytes and placing randomly shaped receptors analogous to an individual lottery ticket with an individual number combination on their surfaces. Okay, let me say that again. The chances of locating a helper T cell with a correctly shaped receptor, even though the body has never seen the potential threat, is actually very good, because before you're infected, your immune system is preparing to win the lottery drawing by manufacturing an army of B and T lymphocytes and placing randomly reshaped receptors on their surface. Current estimates of the number of randomly shaped receptors that your immune system can build. In other words, estimates of the number of unique lottery ticket combinations that it can make is at least 10 raised to the 15th power. That's a huge number, folks. Okay? So what I'm saying is that your body, before you're ever infected with a virus, is trying to ensure that it can win any future battle with an infectious agent by placing randomly shaped receptors on the surface of those cells, so that presumably we will have a helper T cell, like this one here on the screen, letter B, that will have this receptor that can dock to this piece of antigen the very first time it infects the body. Okay, question coming through. Am I making sense? Okay, all right. I hope that I'm getting through to you. This is a difficult thing. It would be easier if I was with you folks in the classroom. Okay, all right. So let that rest with you for a second, and read those notes very carefully so they appreciate what we're saying. All right, I only have about 15 minutes with you, and I need to wrap this up because we need to get into the next chapter, if not today, then the very first thing next class period. So let's say that a helper T cell does bind to one of the pieces on the surface of the macrophage. If and when that happens, right? If and when that happens, your immune system turns on. So next, once the immune system is activated, there will be two simultaneous, well, let me back up. Once the immune system is activated, there needs to be an amplification step. Okay? Because if we think about it on the PowerPoint drawing, this cell right here at letter B is the correct cell capable of recognizing the enemy. But it's only a single cell. And if I've got 50 million virus particles in my body, there's no way this individual cell can fight them all off. So what has to happen next is that this cell, has to go and reproduce many, many times. Because I want to build an army of identical cells that all have this receptor that my pointer is at right now, my cursor is at right now, that will all be able to recognize the pathogen. This process of making many, many cells is called clonal selection. And clonal amplification. We have, in fact, identified the proper helper T cell that can recognize the enemy. Now I've got to clone it. Now I've got to make many, many, many copies of it. This is talked about, folks, on figure 1310, page 248 of your textbook. Page 248, figure 1310. Okay. So the amplification step means the clone well, let me say it this way. The helper T cell that can specifically identify the pathogen is called a clone. This clone has been selected, i.e., clonal selection has taken place. Now, this clone must reproduce many times, clonal amplification, to build an army of cells that can all recognize the same enemy. Okay, and perhaps an inappropriate analogy, but I'll use it anyway. If any of you are fans of Star Wars, those white paratrooper guys, the stormtroopers, aren't they in the original Star Wars mythology? Aren't they all clones of each other? That was the whole point of the Clone Wars in Star Wars, if you're a fan of Star Wars movies. So if you want to pass the next test, go watch every Star Wars movie and it will help you, okay? So at any rate, we need to build a clone army, 1 billion copies of a cell that has the correct receptor to identify the pathogen. That has to happen. That's step H. Any questions, gang? Okay, so we will assume that clonal selection and amplification has occurred successfully. Next, the immune system continues its fight along 2 battlefronts at the same time, okay? I can't talk about them at the same time, so I'll talk about one versus the other, all right? So you're going to have antibody-mediated immunity take place, okay? Antibody-mediated immunity is described for you in your textbook. Now let's see here. All right, page 247 of your textbook, figure 13.8. You have a flow chart there. If you look in the middle of the flow chart on the left-hand side, you see antibody-mediated response. And on the corresponding location on the right side, you see cell-mediated response. This is where I'm at in the conversation right now. So I'm at the antibody-mediated response on the left side of page 247. Okay, the antibody-mediated response is carried out by activated B cells. This means B cells that can recognize the enemy, recognize the virus in this case, okay? The activated B cells will grow and reproduce and specialize into they will specialize into plasma cells. Okay, the plasma cells are a specialized form of activated B lymphocyte. And what you need to know is that the plasma cells are capable of manufacturing antibodies. Okay. These are the proteins that can mark the pathogen for destruction, and you'll read about them in your textbook. You know, there's a lot of conversation in the news these days about antibody testing in association with the coronavirus pandemic, right? We need good antibody tests to see if people have been exposed to coronavirus and whether they potentially are now immune to future coronavirus infections, which may or may not be true. So there's a real big need right now to get accurate antibody tests out into our communities. And, you know, right now things are not so good along that front. There's a lot of work that has to be done. Do you have questions right now? This is a pretty intense topic, and I'm afraid that I'm kind of like overwhelming you with so much at one time. You're really going to have to think about this, gang. Read through the notes and think about it and read through the chapter. Okay, so the activated B cells will become plasma cells, which make your antibodies, okay, which are proteins that mark the pathogen for destruction. Maybe we'll put that down. Proteins that mark pathogen for destruction. And there will also be memory B cells. Okay, memory B cells are cells that live a long time. They float through your body over the next 10 years or so. And if your body is unlucky enough to become exposed to the exact same pathogen, those memory B cells can identify the pathogen very rapidly and typically clear the body of the infection before you ever get sick. Okay, so these are long-lasting cells that live maybe 10 years or so and provide rapid protection if the same pathogen is seen in the future. Okay, so that's carried out by what is termed antibody-mediated immunity, which is achieved with the activated B cells. At the same time that that is occurring, you're going to have a second battlefield, a second battlefront occurring, and that's going to be your cell-mediated immunity. Okay. The cell-mediated immunity is carried out by activated T cells. Okay, and this means T cells that can recognize the virus. Okay, you didn't expect anything with the immune system and all the stuff with coronavirus to be easy, right? I mean, it's pretty intense stuff, and there's a lot of complicated issues going on right now. All right, but let's finish our note taking. The cell-mediated immunity is carried out by activated T cells. The activated T cells will grow and reproduce and specialize into subsets of cells that are also given special names. One of them is an effector cytotoxic T cell. Okay. Effector cytotoxic T cells, if I was going to use an analogy for what they are capable of doing and how they respond, cell-mediated immunity, by the way, is on figure 1313 on page 251. Figure 1313, page 251. The cytotoxic T cells are very similar to an assassin. And I mean that literally. Assassins go out and kill people. Cytotoxic T cells, right? The phrase has the word toxic in it, right? Cytotoxic T cell. These cells are kind of like assassins that go out and they will attack virally infected cells and kill them so that the virus doesn't have a way to reproduce itself. Or they will go out and attack bacteria that are making people sick and attack the bacteria and kill them. So think of the effector cytotoxic T cells as hitmen or assassins that kill virally infected cells and or bacteria. Okay, so one of the ways that your book mentions that they do this is listed on page 251. You'll notice on figure 1313 on cartoon bubble 6B as in boy, 6B as in boy, that there's this helper T cell, excuse me, no, there's this effector cytotoxic T cell that's bound to its target, and they show you a cartoon bubble where they magnify into that region, and you notice there's a substance called perforin that's released. Perforins are molecules that perforate the plasma membrane of infected cells so that the cell dies. So think of the perforin that you see on page 251 as like a hail of bullets from a machine gun or a hail of throwing stars from a ninja. And what we're doing is poking holes into the compromised cell so we destroy it so it doesn't make us sick. Releases perforins to kill virally infected cells. Okay? All right. Now, at the same time that we have effector cytotoxic T cells, we also, in fact, have memory T cells. Okay, your book calls them memory cytotoxic T cells. And in a similar manner, these are also long-lasting cells. They might live a decade plus or minus a few years. And if your body encounters the same pathogen in the future, that cytotoxic T cell can defend the body very rapidly before you even have symptoms of illness develop. All right. So there's a lot that was said today. And I could say more. I don't think I should say any more. So let me open up the floor to you. What questions do you have about the immune system at this time? Is there anything I can try to clarify for you or to make sense of for you? Do any of you need smelling salts because you've passed out from overload? Okay, everybody's quiet. All right, gang. So I have 1 minute left, and there's no way I can even begin the next chapter on this. So I'm gonna call it quits here. I'm gonna take about a 10-minute break, and I will be back on the Zoom channel here. If you need my attention about anything, if you have questions about anything, I'll be happy to help you. Otherwise, if there's no questions, please prepare to do chapter 14 on Wednesday's class. I'm going to have to make it a short abbreviated lecture and cover certain ideas with you so that you're ready to take your lecture exam next Monday. So one final time, do you have any questions I can help you with? Okay, gang. So I'll be back here in 10 minutes. If I don't see you, stay well, stay safe, and take care of yourself and your families. That's the most important thing right now. So I'll see you soon. If I don't see you, I'll see you on Wednesday. So see you later. Take care.